Hello guys, hope that you're good. Today I'm going to talk about the complement system. The complement system is comprised of different types of proteins which are present in our body in the form of pro-enzymes. Or pro-enzymes are not active, but they can be activated by different other molecules that we would talk about in the case of these different complement proteins, that how these are activated. So far, the research tells us that there are more than 30 complement proteins present in our system, and they are complementing our immune system. They are named the complement factors or the complement proteins because they complement our immune responses, or they just enhance our immune responses. The immune response says which are functions or immunity which is carried out with the help of different types of immune cells is enhanced in the presence of these different types of complements. Mainly, these complements are synthesized in the liver, but other immune cells, just like the macrophages and neutrophils, etc., these cells can also synthesize different types of complements in our body. Now, as I told you, these are not active, and these complements need activation. And for activation, there are different mechanisms. Um, uh, we would talk about these mechanisms: the classical pathway, the lectin pathway, and alternate pathway. These are three different ways in which the complement proteins can be activated. But first, let's discuss this question that uh, why activation of these complements is necessary, and why are the complements in the first place important for immunity? Because we say that the complements are enhancing both the innate and adaptive immune responses. It means that the cells of the innate immune system, just like the macrophages, neutrophils, basophils, dendritic cells, etc., and cells of the adaptive immune responses, just like the B cells and T cells, etc. Those cells are involved in adaptive immune responses. So the functions of all these different types of cells is complemented or enhanced by these different types of complements. So how the immune system is making use of these complements? So the immune system is making use of the complements in a similar way as it would use an antibody in some cases. For instance, an antibody is used by the immune system to label different types of antigens or bacteria or other microbes that enter our system. So once they are labeled with an antibody, it's easy for the cells of the innate immune system, for instance, to capture and internalize that particular pathogen or bacteria, because there are receptors present on antibodies. The FC receptors present on the immune cells for antibodies, the FC receptors, and FC for which are for the FC region of the antibody. So when the antibody is complexed with an antigen, this complex can be internalized, because it would attach with the FC receptor of the immune cell, and the immune cell would really or easily internalize this particular complex. So in a similar way, if a particular antigen is complexed with an antibody and a complement comes around and binds to the antibody as well, so the complements also have receptors on the immune cells. So on one hand, there would be a receptor for an antibody, and on the other hand, there would be a receptor for a complement. So it would increase the intensity of this binding of the complex with the immune cell, and in this way um, the that immune complex, this antigen antibody complex, would be internalized more rapidly as compared to if it were only bound with the antibody. So for opsonization or labeling, opsonization is labeling of antigens with antibodies or complements. That is the process in which these complements are involved. That's not the only process, because the complements are also involved in direct killing of the microbes through this membrane attack complex that I would talk about with how this complex is formed. And also some of these complements are acting as enophyllotoxins. Others are used as chemoattractants. So different types of complements are used for different purposes. Now, how these complements are activated because these are pro-enzymes in our blood. The first, and according to the classical pathway, the antigen-antibody complexes, which are formed inside our body, can activate, the complement system. Or, the first complement, which is known as C1, can be activated by the antigen-antibody complex. How this complex is formed when the antigen in the form of certain bacteria, for instance, it enters your body and it is complexed with an antibody which is has been raised against a particular antigen. So the antibody is going to attach with this particular antigen and make a complex. So this complex, once detected inside the body, is a message for C1's hydrolysis or the breaking of C1 into, activation of C1, sorry. And what is C1? This is the first complement, C1. And C1 is comprised of three different proteins, C1q, C1r, and C1s. These are three different proteins, and collectively we call them C1. C1q, this protein, again, a number of small peptides together to form C1q, or this protein. C1q binds to the Fc region of these immunoglobulins, IgM or IgG4 immunoglobulins. In their Fc region, you would find that, during this process of activation, C1q is the first to bind there, followed by C1r and C1s. C1q, once bound to the Fc region of the antibody, activates these C1r and C1s. And once C1s is activated, this C1s peptide goes there and it hydrolyzes the C4 and C2 are these complements. The C4 complement and the C2 complements are, split into parts. A smaller part which is known as A, and a bigger part which is known as B. The B, you can take B for bigger part. So C4, when C1 is activated, C1 binds to the antigen to the Fc region of the antibody, and C1 is comprised of different parts. C1q, binds first, followed by others, and then when it is, it activates C1s, it's going to split C4 and C2 into a smaller part, which is called A, and a bigger part B. So these bigger parts, B, of both C2 and C4 combines to form an enzyme which is known as the C3 convertase. So C4b, bigger part of B, and C2b, the bigger part of C. They would just combine to make an enzyme C3 convertase. And this C3 convertase is responsible for hydrolysis of the complement 3 into two parts, which one is C3a and the other one is the bigger part, C3b. C3a is the smaller part. Now, this C3b, the bigger part of this C3 complement, it again join hands with C4b, C2b, the C3 convertase, to make another convertase, C5 convertase. So C5 convertase would have C4b, C2b plus C3b. And this C5 convertase, what is the function of this enzyme? This enzyme is going to split another complement, which is C5, into a smaller part C5a and a bigger part C5b. And this C5b, the bigger part, which came out of this C5's hydrolysis, is going to complex itself with the rest of the complements, which is C6, C7, C8, C9. So first C6 would bind, followed by C7, then after some time C8, and then C9, which is a group of 12 different peptides that are going to bind with this complex. So it would make a huge complex from C5b to C9, and this complex is known as the membrane attack complex. This complex, just attaches to the membranes of bacteria and burrow holes in them and just bring out the lysis of these bacteria. So that is cytolysis or direct killing of the pathogens that these complements actually can do. So this is one pathway, the classical pathway, which is triggered by this particular complex, but there are other pathways as well. For instance, these particular complements can also be activated by the alternative pathway. According to the alternative pathway, the pathogen the cell membranes have certain types of endotoxins. Some of the bacterial membranes, they have endotoxins which can activate the alternative pathway, or activate the complements through the alternative pathway. Or certain types of parasites can also bring about activation of the complements. So it would bring about spontaneous hydrolysis of the complement 3. The alternative pathway starts with complement 3. It would just bring about hydrolysis of complement 3 into complement 3b, or only the bigger part is formed here, or this complement 3b is attached to the microbe, and along wherever C3b is attached to certain microbe or bacterium. So another factor, which is known in factor B, comes and join hands with him, and it also attaches to the microbe. Once it is there on the surface of the microbe, there's another factor which is known as factor D. This is also a complement. B is also a complement. So factor D would just split this factor B into a bigger part which is known as BB, and a smaller part which is known as BA. So this bigger part BB would combine with C3b to make an unstable C3 convertase, which is known as C3b BB. The bigger part of the complement 3 and the bigger part of factor B are making this unstable, C3 convertase. So this is an unstable C3 convertase of the alternative pathway. So how can it be stabilized? Then another factor, which is known as propidin. Propidin binds to this unstable C3b BB convertase to stabilize it. And once it is stabilized, then this convertase would bring about its function. For instance, it would cleave the complement 3, just like here. This convertase C3BB along with propidin would bring about, the hydrolysis of C3 into the smaller component C3A and C3B. So this when it is cleaved into, the C3 is cleaved into a smaller part and the bigger part. This bigger part, C3b here, or you can also see here, it would just join with this sequence C3b plus C3b. So this is actually this is the C5 convertase enzyme. Now C3 has been hydrolyzed, C5 convertase has been made here. This is the alternative pathways, C5 convertase, which is going to do the same function, to split C5 into a smaller unit and a bigger unit, and then the bigger unit would just combine all other remaining units from C5 through C9. These all bigger units would come together to make the membrane attack complex and bring about the lysis of the bacterial cells. Yet there is another pathway which is known as the lectin pathway. So lectin, or mannose binding lectin that we know are present in our bodies. These are present in our system naturally. They are produced in the body. And these proteins are specific for binding with mannose. So where do you find mannose? So in certain bacterial species, for instance, Neisseria, Salmonella, Typhimurium, Candida species, etc. In the cell walls of these bacteria, you can find different types of sugars like glucose and mannose. So mannose, which is present in the cell wall of these different types of bacterial species, bind to the lectins. And when mannose is bound to the lectins, this actually activates an enzyme which is a mannose associated serine protease 1 and mannose associated serine protease 2 enzymes. These are two different types of enzymes which would be activated by the binding of lectin or with mannose. So this MASP1 or MASP2 proteases, serine proteases, would do the same job as C1s is doing here. It's going to, hydrolyze C4, the complement 4 and complement 2, to make the smaller part A and the bigger part B. And then these parts would combine to make C3 convertase, just like here, it will be the same sequence here. C3 convertase would do the same job, hydrolyze C3 into parts, and the bigger part of this would join this sequence to make a C5 convertase. And C5 convertase again is going to hydrolyze C5 into C5a and B and make this membrane attack complex ultimately. So that's how the membrane attack complex is made. And as I told you, there are different purposes of the complements in our bodies, are different functions of the complements in our bodies. As I told you, some of the complements are anaphylatoxins, for instance, C4a or C3a. The smaller components that we are talking about are C5a. These are also working as anaphylatoxins in our body. C5a particularly also acts as a chemoattractant. So, and if you look at the structure of the mast cells or some other cells, you can find receptors for C5a. C5a or C3a can also bring about degranulation of the mast cells and hypersensitivity reactions. You have studied about it. So this is a direct killing or leveling of the antigens antibody complexes with different types of complements. So now there is a question whether the body can or the complement system can work without immunoglobulins. What type of immunoglobulins actually bind to these different types of complements? IgM and IgG4. These antibodies mostly, bind to these different types of complements. In the FC region of immunoglobulin G or M, these complements would bind. And among the four different subclasses of immunoglobulin G, IgG4 mostly is involved in making these complexes with different types of antigens. In some cases, IgM is also, a powerful activator of, the complement system. So if there are no immunoglobulins, whether it is possible for these complements to work or to be activated. So of course, if you look at these different pathways, among these three different activation pathways, only one pathway is dependent on an antibody, you know. But these two pathways, the lectin pathway and alternative pathway, are not dependent on the antibody. So it means that these pathways can be activated or the complements can be activated even if there is no antibody there or no immunoglobulin in the system. So the deficiency of these complements can also cause various problems. For instance, there is a tightly regulated system in the body in order to regulate these different complements. For instance, C1 inhibitors can regulate C1, but in the absence of C1 inhibitors, for instance, If there are no C1 inhibitors, then what can happen? It can lead to some conditions like, angioedema under, in the deep tissues, or it can also lead to some other types of diseases if the complement system is not activated. For instance, if there is a deficiency of C2, then it can lead to more pyrogenic infections in people who are deficient of C2. There are also a regulatory shift here. C3b is converted again into IC3b with the help of these other factors like factor I and H, which again are regulating each other and regulating the production of C3b as it converts it into IC3b, so that C3b would not be available to make C5 convertase or make the membrane attack complex, because this process has to be stopped somewhere. So this was about the different types of complements and how these complements are involved in complementing or enhancing, our immune responses with the help of these different types of. Okay, thank you.